Hello friends, welcome to a very small session here. We'll briefly talk about a very important technique, the fluorescence recovery after photo bleaching. This is a commonly repeated question in your exam and you should know the very basics of this particular technique. So let's talk about what exactly do we mean about FRAP. So FRAP is a method for determining the kinetics of diffusion through tissue or cells. Okay, so one thing you have to remember here, most of the questions will mention that is used for two-dimensional lateral diffusion, but FRAP is also used for the study of three-dimensional diffusion. Both the studies will be called FRAP only, whether you are determining the two-dimensional diffusion or the three-dimensional diffusion. Okay, and primarily it is used as a means to characterize the mobility of individual lipid or the protein molecules within a cell membrane. And more importantly, it is used for the protein molecules. So we want to identify whether the protein that we are looking at, whether the protein we are studying, is it mobile within the membrane or it is fixed. Most of the transmembrane proteins will be fixed, but the proteins which are in the superficial layer, they are typically mobile. So it will tell us about the nature of the protein, whether it is traversing the whole membrane, and from that we can extrapolate the type of functions in which the protein can participate. So moving on to the exact technique of the FRAP. So what happens? So you can see this is the top view and this is the side view, where here we are seeing the lipid bilayer, all right, and all the protein and the lipid molecules, you can see they are labeled and they are tagged with the fluorescent particle. You can see the red color, this is the fluorescent particle, and we can see the surface. Then subsequently what we do the photo bleaching. When we do the photo bleaching, these tags, fluorescent tags, they will get destroyed, so that now we get a dark surface on the surface. Okay, now if the lipid particles or the protein particles are mobile, then through the movement of the particles, some of the photobleach particles will move out, some of the other particles will start coming in, so that you can see this photobleached part gradually becomes less photobleached and gradually the fluorescence is reappearing. And after some time, when a lot of new proteins have come in, most of the photobleach proteins or the lipids have moved out. The picture is similar to what was there in the beginning. Obviously, some of the photobleach particles will still be there, so it is not as brightly fluorescent as in the beginning, but in the course of time, it may become as fluorescent as the other zones. On the other hand, if this area contains certain proteins which are transmembrane, a big protein or multiple proteins which are transmembrane, which are not mobile. In that case, what will happen? The part which has been photobleached will persist for a longer time, and in fact, it can be fixed. It may not disappear even till the end. So, as you can see here, basically, based on the movement, the recovery of the fluorescence will occur. That is why it is called fluorescence recovery after photo bleaching. The name is fluorescence recovery after photo bleaching. This tells us about the movement of the lipids and the proteins in the lipid bilayer. What you have to know that this is also being used nowadays in the lab in the artificial membranes that we are making for the commercial purpose, for research purpose, and it will help us to identify that okay, the artificial structure that we have synthesized, how it will behave under different conditions, whether the particles are mobile within the layers, can they move from one surface to other or not. So these are the things which we'll get to know from fluorescence recovery after photo bleaching. Coming in your exam again and again, quick look at the basics to help you out in the exam. Thank you very much.